Ladies and gentlemen, the event will begin shortly. We kindly ask you to obey the rules during this event. Specifically, participants are required to dance neatly and politely. Participants are required to turn on their camera and turn off their microphone. Participants are recommended to set their Zoom meeting to speaker view throughout the event. Participants should not carry out any activities that are not related to this event, and participants should avoid any kind of distraction that could disrupt the course of this event. Your cooperation and patience are highly appreciated. Before we move to our next agenda, I would like to remind you to fill the attendance of the 4th ISPST 2020 on link which shared via group chat Zoom. Thank you. The Honorable Dean and Vice Dean of Faculty of Pharmacy Universitas Padjadjaran, Professor Tomaya Wehara, Associate Professor Ponchairo Sitisak PhD, Profesor Dr. Yahdiana Harahap, Dr. Nyimakar Saptarini, Mr. Holis Abdul Holik PhD, Dr. Alia Nur Hasana, all professors, faculty members, distinguished guests are participants present today. Welcome to the 4th International Seminar on Pharmaceutical Science and Technology 2020. Ladies and gentlemen, first of all, let us extend our gratitude to the Almighty, whom without His grace, we will not be able to come together in this wonderful event. The 4th ISPST is a continuation of the 3rd ISPST, which had been held in 2018. This seminar will give the latest findings in areas related to pharmaceutical science and technology. Today, we have more than 100 participants, students, industrial experts, and professionals joining the seminar to discuss and share their knowledge. On behalf of the organizers, I would like to express my gratitude to the presence of all guests, speakers, and participants in this seminar, and a warm welcome to Universitas Padjadjaran. Next, we are going to sing Indonesia National Anthem, Indonesia Raya. To bless this event, may I invite everyone to invoke the Almighty, and we would like to invite Mr. Govarana Wilar, PhD, for the invocation. With all due respect, we now give the time to Mr. Govarana Wilar, PhD.
Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Before we start this seminar, let's pray to Allah subhanahu wa ta'ala. May Allah bless this seminar. Amin ya Allah ya Rabbal Alamin. Hamdan syakirina wa hamdan na'imina hamdan yuaniya ma yukafi majida. Ya Rabbana walakal hamdu kama yanbaghi li jalali wajhikal karimi wa azimi sultanik. Allahumma salli ala Sayyidina Muhammad wa ala ali Sayyidina Muhammad. Ya Allah, please give us easiness and smoothness during we are performing this seminar. Please shower this seminar by your bless, Ya Allah. Allahumma anfa'na bima 'allamtana wa 'allimna ma yanfa'una. Ya Allah, make useful for me, Ya Allah, what you have teach me on this seminar. Please teach me knowledge that will be useful for me, Ya Allah. Ya Allah, glorify us with the light of understanding. Open the understanding of knowledge and give me some knowledge during following this seminar. Allahumma inni a'udhu bika minal ilmi la yanfa'u wa min amali la yurfa'u wa min du'ai la yustajab. Ya Allah, remind me what I forgot and teach me what do not understand, Ya Allah. Ya Allah, please accept our doa. Rabbana atina fid dunya hasanah wa fil akhirati hasanah wa qina azaban nar. Wa salamun ala mursalin walhamdulillahi rabbil alamin. May Allah accept our doa. Assalamualaikum warahmatullahi wabarakatuh.
Honorable guests, speakers, participants, ladies and gentlemen. Now we would like to invite the Honorable Dr. Nyimakar Saptarini as the chairwoman of ISPST to deliver an opening speech as well as to reflect upon the journey to create this event. With all due respect, we now give the time to Dr. Nyimakar Saptarini.
Good morning. Assalamualaikum warahmatullahi wabarakatuh. On behalf of organizing committee, I'm honored and pleased to welcome you, Dean of Faculty of Pharmacy, Universitas Padjadjaran, keynote and invited speaker, and all participants to the 4th International Seminar on Pharmaceutical Science and Technology 2020. The scientific content of ISPST 2020 is truly multidisciplinary. It covers all aspects of computer edit and drug design, aspect on industry and control quality, pharmaceutical analysis, current trend in drug delivery technology, pharmacology and toxicology, cosmeceutical, and current technology on drug formulation. Through sharing and networking, ISPST 2020 will provide an opportunity for researchers, practitioners, and educators to exchange research evidence, practical experience, and innovative ideas on issues related to pharmaceutical science. We realize that this event is only a small effort to contribute exchange in research and experience. Therefore, more collaborative work and continuous relationship between researchers and are still needed. The scientific program is composed of 2 keynote speakers, 6 invited speakers, and 96 oral and poster presenters. The selected article will be published in 3 international journal Scopus Index and 1 national journal Sinta Index. The diverse scientific content represents many exciting achievements and also point to new challenges. I would like to express my gratitude to all of you who support this seminar, especially organizing and Faculty of Pharmacy. I believe ISPC 2020 will be a very favorable meeting for all participants. Thank you.
We express our utmost gratitude and appreciation to Dr. Nyimakar Saptarini for her meaningful speech. Without her, this program would never have been realized. The next agenda, speech and opening remarks that will be delivered by Dean of Faculty of Pharmacy, Universitas Padjadjaran, Professor Dr. Ajeng Diantini, MSc. With all respect, we now give the time to Professor Dr. Ajeng Diantini, MSc.
Bismillahirrahmanirrahim. To our distinguished speakers, member of Faculty of Pharmacy Universitas Padjadjaran, all the participants, and ladies and gentlemen, good morning. Assalamualaikum warahmatullahi wabarakatuh. It is a great pleasure to welcome you to the 4th International Seminar on Pharmaceutical Science and Technology 2020, which is organized by the Department of Pharmaceutical Analysis and Medicinal Chemistry, Department of Pharmaceutical and Formulation Technology, and Department of Biological Pharmacy, Faculty of Pharmacy, Universitas Padjadjaran. First of all, I would like to start by wishing you and your family are in good health and safety in this difficult time. As society begins to slowly recover from the COVID-19 pandemic, it is clear that COVID-19 has reshaped the way we live our lives for the foreseeable future. The world is nowadays facing a lot of predicament, which requires joint hands from different stakeholders that are involved in wide range of action for the positive changes. We know in this difficult time, the existence of science, technology, innovation are a must to transform the world. Therefore, this seminar is very important. This ISPST has been started since 2014, attended by hundreds of participants and researchers that were not only come from Indonesia, but also from other countries. However, due to pandemic situation, the 4th International Seminar on Pharmaceutical Science and Technology will be held through a webinar. This time, the seminar will focus on future perspective in the transformation of pharmaceutical research into applicable industry, with the topic of nanoparticles, radioactive, natural product, and many other interesting topics of pharmaceutical research that will be discussed roughly in these 2 days. On this occasion, I would like to express my appreciation and thankful to all the speakers. They are Professor Tomoya Uehara from Japan, Dr. Din Huang from Australia, Associate Professor Ponchai Rotsitisek PhD from Thailand, Mary Joanne from Philippines, and several speakers from Indonesia. There are Professor Dr. Yahdiana Harahap, Bapak Raymond Chandrawinata PhD, Dr. Nyimekar Saptarini, and Dr. Sri Widodo. I also would like to thank all the participants of the seminar who are coming from many other places. The seminar will serve as a media for researchers, professionals, and students that have interest in pharmaceutical science and its related fields to build collaboration among them. I hope the seminar will accomplish all its aim and earnestly desire that all participants will be able to benefit from presentation and discussion. Last but not least, I would like to say many thanks to all the organizing committee that supervised by Dr. Nyimekar Saptarini for their tremendous effort to arrange and manage this special event. I wish all of the speakers and participants will gain a lot of benefit and insightful experience. Ladies and gentlemen, with saying Bismillahirrahmanirrahim, I open the 4th International Seminar on Pharmaceutical Science 2020. Have a nice seminar and wish you luck. Billahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh.
We would like to express our utmost gratitude to the Honorable Professor Dr. Ajeng Diantini MSE to remarks the opening speech of 4th ISPST 2020. Honorable guests, speakers, participants, ladies and gentlemen. Before we move to our next agenda, I would like to remind you to fill the attendance of the 4th ISPST 2020 on the link which shared via group chat Zoom. Thank you. The next agenda is keynote speech from Professor Tomoya Uehara that will be led by Mr. Holis Abdul Holik, PhD. And before that, I'll read the curriculum vitae of our moderator. Mr. Holis Abdul Holik, PhD, received his doctoral degree from Kiba University, Japan. His main research interest is nuclear medicine, specifically the development of radiolabeling for biomolecules. With all due respect, we now give the time to Mr. Holis Abdul Holik, PhD.
Thank you. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. Thank you for joining this seminar. Today, we will have keynote speech from Professor Tomoya Uehara. For the next 45 minutes, Professor Uehara will talk about chemical design of radiometal labeled antibody fragments for low renal radioactivity levels. Now, I would like to read the curriculum vitae of our keynote speaker. Professor Tomoya Uehara, PhD, was a lecturer in Graduate School of Pharmaceutical Sciences, Chiba University. His expertise is molecular imaging and radiotherapy with research interest in nuclear medicine, especially in development of radiolabeling reagents for biomolecules. The overall objective of his laboratory is to develop radiolabeled molecular imaging probes that provide high target to non-target ratio. To Professor Uehara, time is yours.
Okay, so I share my presentation. Okay. Can you see my presentation?
Yes, we can see.
Okay, so let's start my presentation. Okay, so thank you, Dr. Horis, for kind introduction. So I'm Tomoya Uehara, Chiba University. So now I'm professor in Chiba University. So next year I become a professor. So, and thank you so much for inviting me to this seminar and giving me opportunity to present my recent work. And I'm very sorry that I could not go to London due to the COVID-19. But I'm very happy to present my recent research via web seminar. Thank you to all staffs. And I met Dr. Horis for the first time at this seminar, maybe 2014, I think, second seminar. After my presentation, yeah, fortunately he was interested in my research and he came to our laboratory as a Dr. Ko student. Yeah, he studied so hard and he's a good student and he graduated my laboratory. So I'm good to I have a memory for him. So now I'm very happy to present my research in front of him in this seminar. So now I'm Chiba, so in Japan. So do you know, oh, too slow, okay. then where is Chiba? So this is Indonesia, and this is Japan. So Chiba is middle of Japan, east of Tokyo. And Chiba University is middle of Chiba Prefecture. Chiba Narita Airport is an international airport. Is in Chiba. So you can come here from Jakarta, almost 8 hours. So I think it is not far from your countries. So you look at the shape of this shape of the prefectures. What does you like? So it is dog. Okay. Oh, so slow. Oh, sorry. So sorry. This is dogs. That is not a dog. Just fairy. This is Chiba-kun, our prefecture's mascot. So if you come here, you can see this mascot. So many places, I think. Okay. So now today I talk about chemical design of gallium level antifragment for low linear activity levels. Okay. First, I introduce molecular imaging techniques using radioisotope. The radionuclides, such as the gamma ray emitting radionuclides and beta plus emitting radionuclide combined with molecules such as antibody fragment, peptide, and so on. And then produce radio-label compound. When radio-label compound injected into the subject, these radio-label compound accumulated in some tissue as some receptor or enzyme and so on. A gamma ray emitting from radionuclide in radio-label compound detected by gamma camera, such as PET and SPECT. From these, we can know the cellular function or enzymatic function and physiologies. Oh, so slow. Sorry. Oh, okay, so this slide shows the future of PET. PET is positron emission abbreviation of positron emission tomography. A carbon 11, nitrogen 13, oxygen 15, fluorine 18, and gallium 68 are represented radionuclides for PET. A positron emitting from radionuclide combined with electron, and then two annihilation radiation were produced. A PET system detected these two annihilation radiation coincidentally, and then provides 3D images. Usually, PET system is higher sensitivity and provide more quantitative image compared with SPECT. On the other, however, half-life of these radionuclides is so short, from 2 minutes to 2 hours. Therefore, a cyclotron is required to use these radionuclides. So this slide shows the future of SPECT. SPECT is the abbreviation of single photon emission tomography, and the 99 technetium, iodine 123, gallium 67, and indium 111 are representative radionuclides for SPECT. The gamma ray emitting from radionuclide is detected by gamma camera. SPECT imaging by acquired from the gamma camera to multiple sight from multiple angles, and then provide 3D images. The half-life of these radionuclides is longer than that of PET radionuclides. Therefore, we can pursue the radioactivity for a long time. And these radionuclides is available from commercially. From these, the number of SPECT machines is much higher than that of PET scans. In Japan, the number of SPECT machines is almost 5 times higher than that of PET machine. Okay, so recently, there are so many kinds of antibodies against various antigens that have been developed. Therefore, if we label antibody by radionuclide, we can image the various molecules. However, the blood clearance of radiolabeled antibody is so slow. Usually 2 or 3 days required to obtain clear image. In case of radiotherapy, it causes hematologic. From these, recently, antibody fragments, such as FAB fragment, FB fragment, have been developed. These antibody fragments show faster blood clearance and more uniform distribution compared with whole IgG. Therefore, these are good candidates for the carrier of radionuclide. This slide shows the conventional preparation method of radiolabeled antibody fragment. Bifunctional chelate, such as para-ethylene benzyl noter, conjugate to FAB fragment by an amine group of epsilon group of lysine, and then produce bifunctional chelated noter bifunctional chelate conjugate FAB. In this case, a noter FAB. So this noter FAB was labeled by gallium citrate or gallium chloride because noter is a good chelate for gallium, and then produce a gallium noter FAB. So this slide shows the SPECT-CT image of gallium noter FAB in tumor-bearing mice. So this image was obtained by small animal SPECT-CT system. Chiba University Isotope Center has this machine. The gallium noter FAB visualizes tumor very clearly. However, as you know, renal radioactive level also observed. In general, radiolabeled antibody fragment show high renal uptake. It causes decreased diagnosis accuracy and increased side effects of radiotherapy. It is a big program to use these radiolabeled antibody fragments in clinical stage. So this slide shows the retention mechanism of radioactivity in the kidney after injection of RI-labeled peptide. When RI-labeled peptides were injected into the body, and then this peptide is filtered at glomerulus in the kidney. After that, this radio-labeled peptide is reabsorbed into the proximal to the cell and then metabolized in the lysosome, then produce radiometabolite. So emission rate of these radiometabolite from kidney to urine result in retention of radioactivity in the kidney. In case of granulota FAB, granulota lysine is a final radiometabolite. So this granulota lysine is retained in lysosomal fractions. From these considerations, it is suggested that if these radiometabolite could be excreted from lysosome to urine rapidly, renal radioactivity level would be reduced. On the other hand, at 1997, NIH group, National Institute of Health in the United States, that group reported that granulota labeled distributed stabilized FV were injected into mice. They found two radiometabolites, granulota lysine and granulota methionine, were found in the kidney. Because the N-terminal of this distributed stabilized FV were methionine, so they found this radiometabolite, granulota methionine. When they analyzed the clearance of this radiometabolite from the kidney, they found that granulota methionine was excreted more rapidly than granulota lysine. So after 6 hours, they found granulota lysine were found in the kidney, but granulota methionine was disappeared. From these findings, we tried to develop a granule labeling agent that liberates granulota methionine by lysosomal proteolysis of antibody fragment. From this consideration, we designed a granule labeling agent, NOTA-MI-marimide. This reagent consists of 3 parts: chelating moiety for gallium, that is NOTA, and the metabolizer linker, methionine isoleucine. And the third part is the binding moiety for antibody fragment, marimide. In this case, we selected methionine isoleucine sequence as metabolic linker because amino acid next to methionine in the previous distributed FV reported NIH group was isoleucine. So we selected this sequence. If methionine isoleucine sequence would be cleaved by lysosomal enzyme, NOTA methionine would be produced. So this slide shows the preparation method of NOTA-MI-marimide. A para-ethylene NOTA conjugate with methionine isoleucine ethylene diamine marimide to produce NOTA-MI-marimide. And this slide shows the conjugation method of NOTA-MI-marimide to FAB fragment. The FAB was cyclated by iminocycline and then conjugated to NOTA-MI-marimide to produce NOTA-MI-FAB. As a control compound, we synthesize NOTA-FAB. In this case, para-ethylbenzyl NOTA was conjugated to FAB fragment directly. So this slide shows the labeling of NOTA MRI FAB and NOTA FAB and the stability assessment of GALU NOTA MI FAB and GALU NOTA FAB. The NOTA conjugate FAB was labeled by gallium in acetate buffer at 37 degrees 40 minutes, and then produced a GALU NOTA conjugate FAB. The reactant chemical ease of GALU NOTA MI FAB was 95%, as same as GALU NOTA FAB. And GALU NOTA MI FAB was stable in micellar for 24 hours, as same as GALU NOTA FAB. So this slide shows the biodistribution of radioactivity in normal mice after injection of GALU NOTA MI FAB and GALU NOTA FAB. The red one is GALU NOTA MI FAB and blue one is GALU NOTA FAB. The blood clearance of radioactivity was similar between GALU NOTA MI FAB and GALU NOTA FAB. So GALU NOTA FAB shows high linear activity level from 1 to 6 hours. On the other hand, GALU MI FAB shows significantly lower linear activity level from 3 to 6 hours. And GALU NOTA MI FAB shows higher urinary excretion than GALU NOTA FAB. So this slide shows the reverse phase HPLC analysis of kidney homogeneous after injection of GALU NOTA MI FAB and GALU NOTA FAB. Low-risk column is a standard compound GALU NOTA methionine and GALU NOTA lysine. After injection of GALU NOTA FAB, GALU NOTA lysine were observed in the kidney both 1 hour and 6 hours post-injection. On the other hand, after injection of GALU NOTA MI FAB, GALU NOTA methionine was observed at 1 hour post-injection. But after 6 hours, GALU NOTA methionine was disappeared. So this slide shows the radiometabolite in urine after injection of GALU NOTA MI FAB. The size exclusion HPLC shows that 95% of the radioactivity was observed in the low molecular weight fractions. The activity in the low molecular weight fractions were analyzed by reverse phase HPLC. Chromatogram chart shows that GALU NOTA methionine was the major radiometabolite in the urine. So these results indicate that the GALU NOTA MI FAB was deabsorbed into the kidney, and then this was metabolized in the lysosome, and then produced GALU NOTA methionine. So this GALU NOTA methionine was excreted to urine rapidly, and then renal radioactivity level would be reduced. So this slide shows the biodistribution of radioactivity into the tumor-bearing mice. Tumor to muscle ratio of GALU NOTA MI FAB is similar to those of GALU NOTA FAB. However, tumor to kidney ratio of GALU NOTA MI FAB were much higher than those of GALU NOTA FAB and GALU NOTA FAB, both 3 hours and 6 hours post-injection, almost 2 or 3 times higher. So So these results suggest that the gallium nota MIFAB reduce the renal radioactive levels without impairing the tumor radioactive level due to the release of gallium nota methionine by the lysosomal proteolysis. However, in these strategies, several hours were required. In this case, 3 to 6 hours were required to reduce renal activity levels. We thought that if urinary radiometabolite can be produced before lysosomal proteolysis, we can reduce the renal activity level more rapidly. From these hypotheses, we propose new strategies, brush border strategies. In these strategies, when the array level peptides are injected into the body and then filtered at glomerulus. After that, this peptide is reabsorbed into the proximal tube cell. At this time, brush border membrane enzyme cleaves the metabolite linker and then produce urinary metabolite. Renal activity level would be reduced more rapidly. From this consideration, we designed and synthesized new gallium labeling agent, nota MVK maleimide. This was also consist of 3 parts. One is a radiometabolite part, a gallium nota methionine. And next part is metabolite double linker, a methionine valine lysine sequence. And the third part is a binding molecule for antibody fragment, in this case, maleimide. In this study, we selected the methionine valine lysine sequence as a metabolite linker because this sequence was reported as a metabolite linker, metabolized by a brush border membrane enzyme. So we selected this sequence. So this slide shows the synthesis method of nota MVK maleimide. The para-ethylene benzyl nota was conjugated to methionine valine lysine maleimide, MVK maleimide, to produce a nota MVK maleimide. To investigate the recognition of enzyme to the MVK sequence, we synthesize low molecular analog, nota MVK benzo. In this case, benzyl group was used instead of maleimide. And this nota MVK benzo was labeled by gallium via gallium acetate, and then produced gallium nota MVK benzo. So this slide shows the recognition of BB brush border membrane enzyme to the MVK sequence in gallium nota MVK benzo. The brush border membrane basic BBMV prepared from rat kidney were incubated with gallium nota MVK benzo at 37 degrees for 2 hours, and then analyzed by TLC and HPLC. After 2-hour incubation, gallium nota methionine was observed. Therefore, these results indicate that the MVK sequence in gallium nota MVK benzo was recognized by brush border membrane enzyme. And the release of gallium nota methionine was inhibited by phosphoramidone. Phosphoramidone is the inhibitor of neutral end peptidase. Therefore, this result indicates that gallium nota MVK benzo was recognized and cleaved by neutral end peptides. This slide shows the stability of gallium nota MVK benzo in mice serum. Gallium nota MVK benzo was incubated in mice serum at 37 degrees for 24 hours and then analyzed by HPLC and TLC. As you know, gallium nota MVK benzo was stable in mice serum for 24 hours. So this slide shows the preparation method of gallium-labeled FAB fragment. FAB was cyclated by immunocycline and then conjugate with gallium nota MVK marimide to produce nota MVK FAB. And nota MVK FAB was labeled by gallium acetate to produce gallium nota MVK FAB. In this study, we synthesized two different compounds, gallium nota MI-FAB and gallium nota FAB. In case of gallium nota MI-FAB, gallium nota methionine was produced by lysosomal proteolysis. And in case of gallium nota FAB, there are no metabolite linker, so precyl benzyl nota conjugate to FAB directly. So this slide shows the biodistribution of radioactivity in normal mice after injection of gallium nota-labeled FAB. The blood clearance of radioactivity was similar among gallium nota FAB and MI-FAB, FVK FAB. However, gallium nota MI-FAB shows lower linear activity level compared with nota FAB, black one, as the same as previous report. On the other hand, gallium nota MVK FAB shows significantly lower linear activity level in the kidney than these either gallium nota MI-FAB and gallium nota FAB. So this slide shows the radiometabolite in urine after injection of gallium nota MVK FAB. The size exclusion HPLC shows that 70% of radioactivity were eluted at low molecular weight fractions. And the radioactivity in the low molecular weight fractions were analyzed by reverse phase HPLC. The gallium nota methionine was a major radiometabolite in urine. So this slide shows the biodistribution in tumor-bearing mice after injection of gallium nota conjugate FAB. The gallium nota MVK FAB was similar radioactivity level in tumor compared with gallium nota MI-FAB and gallium nota FAB. However, gallium nota MVK FAB significantly reduced the kidney radioactivity level compared with gallium nota MI-FAB and gallium nota FAB. But these results indicate that the gallium nota MVK FAB reduced linear activity levels without impairing tumor radioactivity levels. Finally, we obtained a specificity image after injection of gallium nota conjugate FAB in tumor-bearing mice. The gallium nota FAB visualized tumor very clearly, but high linear activity levels were also observed. The gallium nota MI-FAB visualized tumor very clearly, and kidney radioactivity level were reduced compared with gallium-nota FAB, but still high radioactivity were observed. On the other hand, gallium-nota MVK-FAB shows much lower linear activity level and visualize tumor clearly. As a result, the gallium-nota MVK-FAB provides a high contrast tumor image compared with these gallium-nota conjugate FAB. So, summary, gallium-nota MVK-FAB provides a much higher contrast tumor image when compared with gallium-nota MI-FAB and gallium-nota FAB due to the release of gallium-nota methionine by the action of BBM enzymes. So conclusions in this seminar, because antibody fragment and constructs share similar metabolic fate in the kidney, the present labeling procedure would also apply to a variety of antibody fragment and construct of interest. And derivating radio-labeled compound to run excretion from antibody fragment as the linear BBM would be promising strategy to reduce the linear activity levels. So this study was done in our laboratories, and this strategy was developed by Professor Arano, my boss. So now, so the COVID-19, but when the COVID-19 is gone, so please visit Japan. So I think this is very nice countries for you. So there are many sighting seaport, Hokkaido, Tokyo, Kyoto, Osaka, Hiroshima, and so on. And Chiba is very nice place also. Tokyo Disneyland also in Chiba. Interest my research, please contact me and please contact Dr. Horace. So he knows me well so much in my laboratory. Thank you so much. Thank you, Horace.
Okay, thank you, Sensei, for your such a nice and interesting presentation. And also thank you for introducing me more about me. Actually, we you came to Indonesia, our first ISPST.
Yeah, so it's a good memory for me.
Okay, now we come to discussion session. For the audience who want to ask Uehara Sensei about this particular topics, please raise your hand. Yeah, I think maybe while waiting for the audience, okay. I have a question. In case of you use Nota.
Yeah, in the case we use Nota, yes.
You use Nota, but you use Nota Parisian benzyl Nota.
Yeah.
Why not chelator instead of another Nota chelator?
Yeah, first, yeah, I think Nota is more famous than Nota, but so first, yeah, NIH group so use Nota first. So they found that Nota methionine was good candidate for the radiometabolite. So first I tried to use gallium Nota methionine as radiometabolite. So now, so in our laboratory, synthesize Nota chelate for the radiometabolite. So maybe this year or next year, we publish the data, I think.
I see. Okay. Thank you. Now we have a question from Dr. Porchai, please.
Okay. Good morning, Professor.
Good morning.
Nice to meet you again. I have been to Chiba many times.
Sorry. Okay. Yeah.
I have two questions. The first question is,
Thank you.
for the DOTA compound, do you synthesize para-thiol-cyano benzyl nota in your lab by yourself, or is commercially available?
Yeah, previously we synthesized. I think no, Horace knows so well, I think. But recently we bought the chelate. Yeah, that is also commercially available. United States companies sell that chelate.
I see. And the second one is that I noticed that your linker is kind of specific to the lysosomal enzyme, lysosomal protease enzyme. Do you know if there is any difference between the lysosomal protease enzyme in the normal cell and in the cancer cell?
Yeah, I think, yeah, in lysosomal enzyme, almost same, I think. Yeah. Lysosomal enzyme is, I think, not specific enzyme. I think all peptide or antibody and protein can be metabolized. So I think enzyme is not different between the tumor and normal cell, I think. Both is metabolized, I think.
So in that case, why we see the difference in the tumor size?
Tumor size, I think, yeah. Tumor size, I think in the tumor, I'm sorry. What is your mean of the difference?
I mean the size of the tumor. How we see the selectivity from the muscle to the tumor, or between the kidney and the tumor.
I see.
How selectivity happens between the one with the linker and the one without the linker.
I see. So in the tumor, so peptides bind to the surface of the tumor cell, but in the kidneys, so compounds internalize into the kidney cell and metabolize. But in tumor, peptide bind to surface, not internalize. So in the kidney, radioactivity reduced, but in the tumor is not reduced because not metabolized, I think.
I see. Thank you very much. I understand.
Thank you. Question. Thank you.
Okay, thank you, Dr. Pochai, for the question. And there is another question from Dr. Yan Sopian. I read the question: how to select a properly amino acid that support the to detection method using labeling? Thank you. Yeah, you can read the how to select a properly amino acid that support to detection method.
So how select the amino acid? So amino acid. So sometimes sound is stopped or something. So I think the line is not good now. So
maybe Dr. Yan, can you repeat your question?
Yes, support to detect the method using how to select the properly amino acid that support the detection. Properly amino acid support detection. So that means how do we select the metabolic linkers, amino acid sequence. Okay. So usually we analyze, we check the some paper, and then we combine to curate, and then check the metabolites. But sometimes good, sometimes not good. So.
Okay. Yeah, maybe Dr. Ian.
Mm-hmm.
Yeah, maybe we usually we select. I don't know exactly. Oh, so what is the point? The question?
Yeah, yes, yeah. In this case, we use MVK sequence in this seminar. But yeah, I tried another amino acid sequence also. We tried, but sometimes yeah, that is not good. So that is try and error and something we reform. Yeah, finally we get this sequence. Yeah.
Okay, thank you. Any other question?
Thank you so much.
Please raise your hand so the committee, the co-host, the host can see you.
Yeah. Oh, one, I have a question. Yeah, yeah, I read the chat. Yeah, I also. Okay.
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 Miss Rina Fajri, please ask your question to Wera Sensei.
All right. Good afternoon, maybe Sensei Wera Sensei.
Yeah.
Good afternoon in Japan.
Oh, yeah, almost afternoon. So now
almost afternoon.
30, so.
Yeah, so my name is Serena. I'm the academic staff from Padjadjaran University, but now I'm currently studying in the University of Sydney. So it's already 1:30 now in Australia.
Oh.
Yeah.
Yeah, right.
So it's a really an interesting topic that you presented. So our group here actually also working with antibody, but we conjugate with nanoparticles.
Mm-hmm.
So one of our group members is working with to synthesize a bispecific antibody.
Mm-hmm.
And I'm wondering whether it's possible to label the antibody after we conjugate it with nanoparticles. Whether, well, the purpose will the purpose be the same, or it's another purpose for labeling or conjugating? It's also for cancer therapy.
Mm-hmm.
So this is my first question.
Yeah.
So this.
Okay.
Yeah. So I'm just simply asking whether after we conjugate the antibody with nanoparticles or drugs, maybe, so is it possible to do the labeling with radionuclide?
Yeah, I think, yeah, we can label it, I think, yeah.
So the second question is how we choose the right radionuclide to label our antibody. Like you are using now is gallium,
Yeah.
right? So I'm aware there is another like technetium. So how do we choose the right radioactive compound to label our antibody?
So first we concern with the molecular size of the target compound. If that is big, so blood clearance is so slow, so we use radionuclides that have longer half-life. So gallium-67 is half-life is almost 3 days, but technetium is 6 hours. So for example, so we label the antibody by technetium, but usually we need just 1 day or 2 days to visualize the tumor. But technetium is half-life 6 hours. Already that is disappears, so we cannot visualize tumor by technetium.
So basically, it depends on our target.
Depends on the molecular size. So
okay.
I don't know your nanoparticle size. So if your compound is so big, I think engine 111 or gallium 67 or longer half-life is more available, I think. But the small compound, I think technetium is also available.
Okay. So it depends, actually, it depends on the target.
Yeah, and molecular size also, yes.
Of the antibody, the nanoparticle size.
Yeah.
All right. Thank you. Thank you very much.
Okay. Thank you, Ms. Rina. Yeah, actually, she is also a staff member of my department.
Okay.
Any other question? We still have time.
May I have question, Dr. Halis?
Okay. Yeah, Prof. Muhtraidi, our Vice Dean of the
How are you, Professor Wehara?
Yeah.
Nice to meet you again.
Nice again, yeah.
I'm just nice presentation.
Thank you so much.
Yeah, I'm just wondering about your presentation.
Okay.
You use DOTA and NOTA, right?
Yeah, NOTA. In this case, NOTA is.
Oh, NOTA. Yeah, okay. As we know, NOTA cannot stand in hot temperature, right?
I'm sorry?
NOTA cannot stand in hot temperatures.
Hot temperatures. Yeah.
How many temperatures the NOTA can be synthesized of your compounds?
Yeah, yes, I'm sorry. How many? I'm sorry.
Okay. Okay. NOTA, as I know, NOTA cannot hold in hot temperatures.
What
temperatures?
Yeah, I think NOTA is being good curate and so commercial available and sorry.
Okay. So how do you do this synthesis if it's not NOTA has to be done in hot temperatures?
Yeah, NOTA is synthesis from cyclone, and we can also buy commercial available. So
okay,
yeah, I think.
Yeah. Okay. Yeah. Okay. I have second question.
I'm sorry. Yeah.
Second question. Yeah. As Dr. Ian asked to you about how to select the amino acid property, I have question how to design your ligand. Use can bind to target a protein target like the antibody. So how to design your ligand? Use the ligand can bind against to target. Your protein target.
Yeah. Yeah. So. Yeah, my compound. Sorry. So what is what I'm sorry. I cannot catch the call.
Okay. You synthesize your compound. Use the compound can be interactive with the protein target like the antibody, right?
Yeah.
So my question is how to design your ligands of radiopharmaceutical.
Yeah.
Use these ligands can bind to antibody.
Yeah, antibody. Yeah, but in the antibodies, so that is non-target tissue is a liver, I think, not kidney.
Okay.
Yeah, so yeah, my compound is also reduce the liver activity also. But I think the clearance rate from the liver is not so high. So yeah, maybe we can use that reagent, but the contrast is maybe more later times, I think. Yeah. Not so. In the kidney, so brush border membrane enzyme is already exist, but in the liver, there are no brush border. So that I think same to I think Gavnota MI FAB is a previous strategies. I think that is same. I think. Yeah, we can use that, but not so fast clearance, I think.
Okay, thank you.
Yeah,
okay, Professor O'Hara, I hope I can visit in the next year to your lab.
Oh, thank you.
Thank you for Mutaridi for the question. And also we have one last question since we have time limit from Dr. Yoga Wardana. How to determine the safety of radio labels against genetic mutation that cause cancer?
That is a big problem. So yeah. So I'm not used to the radiation safety. So but usually, so we use a patient with cancer. So we must cure the cancer. So that is not a big problem, I think. We did not use our label compound to normal patients. So I'm sorry. So I do not have a good answers for these questions. I'm sorry.
Okay. Yeah, since recently I also work with nuclear medicine department, and also we usually only tiny amount of radioactive and also we use activity in what is a small amount radioactivity, non-pharmacological effect. Maybe. Then
so
okay.
The radioactivity we use is not so high. So I think genetic mutation is not occurred, I think.
Okay, since we are limited by time,
thank you.
Thank you for your presentation and for such an active discussion today. Yeah, we have to finish this session. Thank you, Wara Sensei.
Thank you so much
for the audience.
For inviting me this symposium. Thank you. So.
Okay, I will give back to the committee.
As a token of appreciation, we would like to invite Mr. Holis Abdul Holik, PhD, to kindly hand over the certificate to Professor Tomoya Uehara.
Thank you, Uehara Sensei. It's an appreciation of your contribution in this seminar. On behalf of the Dean of the Faculty of Pharmacy, Universitas Padjadjaran, and the ISPST committee, I would like to give you a certificate. And this certificate will be delivered by email.
Thank you.
Soon after this seminar finished. Thank you.
Thank you very much, Professor Tomoya Uehara and Mr. Holis Abdul Holik, PhD. The next agenda is poster session. Poster session will be held at this online room. After poster session, the event will continue to plenary session. Thank you. Before we move to our next agenda, I would like to remind you to fill the attendance of the 4th ISPST 2020 on the language chat via group chat Zoom. Thank you. The next agenda is plenary session from Associate Professor Poncai Rositak, PhD, Professor Dr. Yahdiana Harahap, and Dr. Nyimakar Saptarini, that will be led by Dr. Alia Nur Hasana. And before that, I'll read the curriculum vitae of our moderator. Dr. Alia Nur Hasana has research interest in sensor, molecular imprinting, and analytical method development. He has received her doctoral degree from Bandung Institute of Technology. One of her publications is Synthesis and Application of Cleavable Climate Imprinted Polymer for Solid Phase Extraction in Serum Samples using itaconic acid as functional monomer. With all due respect, we now give the time to Dr. Alia Nur Hasana.
Thank you to the Master of Ceremony. Ladies and gentlemen, I'm very pleased to see you here and welcome all of you to the first plenary session on 4th ISPST. The speaker for this session are three of the invited speakers, which is Associate Professor Poncai Rositak from Chulalongkorn University, Thailand, Professor Dr. Yahdiana Harahap from University of Indonesia, and Dr. Nyimakar Saptarini from Universitas Padjadjaran. Before the presentation begins, let me read the curriculum vitae of the first invited speaker. Associate Professor Poncai Rositak. Poncai Rositak, PhD, is an associate professor from Department of Food and Pharmaceutical Chemistry, Faculty of Pharmaceutical Sciences, Chulalongkorn University, Bangkok, Thailand. In 2002, he finished his doctoral degree in Pharmaceutical Sciences from Department of Pharmaceutical Sciences, School of Pharmacy, University of Southern California, Los Angeles, California. Professor Poncai Rositak, Associate Professor Poncai Rositak, PhD, has published journal articles relating to pharmaceutical science and pharmaceutical analysis. He also published books about curcumin. Please, Associate Professor Poncai Rositak, time is yours. You have 30 minutes to share with us your research, and then we will have discussion session with all invited speakers of this session later. I will let you know when the time already 5 minutes left. From the committee, please prepare the PPT file of the Associate Professor Poncai Rositak. Okay, time is yours.
Thank you, Dr. Alia, for your kind introduction, and I would like to thank the committee of the 4th International Seminar on the Pharmaceutical Science and Technology, Faculty of Pharmacy, University of Padjadjaran. It's really a pleasure to have a chance to give a seminar in this meeting. For myself, my name is Poncai Rositak. I'm going to give the seminar on the topic of pharmaceutical applications of growth drugs for enhancing physical-chemical properties, bioavailability, and biological activities of natural products. The research is going on based on the research unit, the so-called natural products for aging and chronic diseases at the Department of Food and Pharmaceutical Chemistry, Faculty of Pharmaceutical Science, Chulalongkorn University. Next slide, please. If you don't mind, if the committee have the pointer, please kindly point out the word that I'm talking along with my talk. Thank you very much. The research unit, natural products for aging and chronic disease. Actually, we have three main research units. The three parts, the first part is the chemistry and biopharmaceutics. This research area involves the natural product chemistry. We do some isolation and structural modifications, and also some structural elucidation. And the second part is the physical-chemical property. We determine the solubility, log P, and stability of the compound to look at the property where it can be enhanced. The last one is the biopharmaceutical. We're looking at the cellular uptake, the drug transport, and the in vitro permeability across the cargo tube that imitate GI epithelial cell. The second part is the pharmacology part. In this part, we do some biological activity testing as listed on the slide. The second one is we also do the in vitro metabolism. And the last one is the in vivo pharmacokinetic. We actually do the pharmacokinetic in rat and mice. The last part is the pharmaceutics area. In our research unit, we develop some drug delivery system, especially the nano drug delivery system, the polymer drug conjugate, and the macromolecular drug conjugates as well. But this one, we are currently develop the antibody drug conjugates as well. But for today, the natural product that I'm going to talk about is the curcumin. For the next slide, we will see the rhizome of the turmeric. Turmeric, the scientific name is Curcuma longa. In Southeast Asia, we have been using this plant as a spice, and we know that curcumin is the major part of the plant. The structure of curcumin is the phenolic compound. You can see the structure. There are two parts, which is a phenol on the left and the right of the compound. And the one that I do the circle in the red circle is actually we call the keto-enol form. So in the structure, they are very interesting because there are many functional groups. For example, phenolic, OH, keto, carbonyl, enolic, OH, and methoxy group. These functional groups are actually responsible for the biological activity of curcumin. And how important of curcumin? If you look through the next slide, our lab has summarized the clinical trials for curcumin as of April 2018 from the website of the clinicaltrials.gov. And this website, whoever wants to do the clinical trial will register to this website, and in our lab, we collect the number of curcumin undergoing on the clinical trial. And you can see that there are many diseases that have been undergo clinical trial with curcumin, as you see on the slide. And I would say that it's nearly several hundred clinical trials going on right now. And if you go to the PubMed.gov, you can see that in the PubMed, there are more than 10,000 publications on curcumin. That means curcumin are very popular for the researcher. And if we focus on the cancer diseases for curcumin, we can go to the next slide. You can see that if we focus on the clinical trials on curcumin for cancer prevention and treatment, you can see the clinical trial is more than 50 clinical trials going on as of 2018. So up to now, probably it's more than that. However, if you look at the dose of curcumin that used for the prevention and treatment of cancer, the dose is very high. It's ranging for several grams to more than 10 grams. You see some of them like 8 grams per day. Some of them is go for 4 grams per day. And this is the limitation of curcumin. That means we need to take a lot of curcumin in order to get the benefit of curcumin. However, in the patient, it's very difficult if you ask the patient to have like 5 grams of curcumin every day. This is very difficult for them. So Now we have to find out why curcumin needs to be taken a lot and why such a high dose is needed to get the benefit of curcumin. So I showed you on the next slide is the limitation of curcumin. And you can see that curcumin have a very, very poor stability. In terms of stability, there are two types of stability. One we call the chemical stability, and the second one we call is a metabolic stability. Curcumin have disadvantages on both stability, have the chemical instability and metabolic instability. On your left-hand side, you can see in the middle is the structure of curcumin. And if you go upward, go to the top, you can see that curcumin can be conjugated by the phase 2 metabolite. And you can see sulfate conjugation and glucuronide conjugation and lead to the inactivation of curcumin. So go down to the bottom, you can see that curcumin undergo phase 1 metabolite. It can be hydrolyzed easily to different compounds. That means it can be degraded to some other compound all the way to vanillin, which is the last one. Okay, from your right-hand side, you can see the phase 1 reductive metabolite of curcumin. Curcumin can undergo reduction and give tetrahydrocurcumin, hexahydrocurcumin, octahydrocurcumin, and some of them have no biological activity. However, some of them has promising biological activity. And because of the instability of curcumin, leading to the problem of the requirement for high doses of curcumin for the prevention and treatment for several diseases. Now, it's up to the point how we can overcome the problem of curcumin. For this presentation, I would like to give an example of using flow drug approach to overcome the stability of curcumin. Some of you may not have heard about the word prodrug. So let me give you an overview on the next slide. What is a prodrug? Prodrug design is the method for develop or improve the drugs that have some problem. For example, in the drug called nevastatin, this drug has a problem, and because this drug has some biological problem, therefore the drug have been developed as a prodrug. And you can see that the prodrug is the ester prodrug on the red. You can see on the red of the left-hand side. That is the ester. You can see the nevastatin and the carbonyl O, which is the ester prodrug of nevastatin. And it actually enhances the absorption of nevastatin. So how the products work, if you look at the picture on the top, the drug actually cannot pass through the barrier. So you can see the drug in the green color is actually go through the GI and then cannot be absorbed. However, if we conjugate the drug with the carrier, which is called pro-moiety, the drug connect to the blue color, which is pro-moiety, eventually the whole molecules can pass through the GI membrane and go inside the cell. It can be the GI epithelium cell or even go to the plasma or go to the specific cell. Then it gets bioactivation and cleave the linkage between the drug and the pro-moiety, and eventually we give the drug. So in this case, we can get the benefit of getting the drug inside the cell or getting it in with the better absorption. There are so many drugs. Besides nevastatin, mycophenolic mofetil also an ester prodrug. Oseltamivir also an ester prodrug. enalapril, cefuroxime acetyl, tafopros. All these are the drugs that use the prodrug design approach to improve the oral bioavailability of the drugs. So with this strategy, we try to use a prodrug approach to improve the oral bioavailability of curcumin. So how we do it? On our next slide, we use the compound, what we call the succinic acid or succinate ester. So we develop the succinate ester prodrug of curcumin. The synthesis of this prodrug of curcumin is to use curcumin conjugate with curcumin on the left-hand side, the structure of curcumin conjugate with succinyl ester, which is the small dicarboxylic acid. Yes, and we use DMAP as a catalyst, and we use dichloromethane as a solvent. And we eventually obtain the prodrug of curcumin. And hopefully this compound can eventually enhance the stability of curcumin and can be readily absorbed through the GI and go to the blood. And then in our body, we will bioactivate the prodrug of curcumin, and we get curcumin to process the biological activity eventually. So this is the concept of how we improve the bioavailability of curcumin using the succinic acid prodrug approaches. So now we have this kind of approach since the year 2011. So if we go to the next slide, what we work on with this compound for the last 10 years, we have do some physical chemical property and biopharmaceutical property evaluation. We have working on the metabolism and pharmacokinetics of this compound. And also we have determined the biological activity, the so-called pharmacodynamic of the compound. So this is an outline of the work we have done, and we have published a number of papers on the curcumin prodrugs, almost like 15 papers. And from the next slide, each slide will represent different publication that we work on. It's very summative, and I hope that you can share this slide to all the students and they can go through the detail how we perform the experiment by going to the detail in the publication. However, for this presentation, I will give a very brief overview, the summation of each publication on the curcumin prodrug. For the first publication we published in the year 2011, we demonstrate that our curcumin prodrug, which is the structure on the right-hand side, it actually have the better actually it has a poor solubility and similar log P to curcumin because we have the ethyl ester. However, if we look at the stability on the left bottom panel, we can see that CDD have a very good stability profile. So basically, curcumin have the half-life about 15 minutes. However, CDD, which is curcumin diethyl disuccinate prodrug of curcumin, have the half-life in the phosphate buffer about 8 hours. So with this achievement, we finally can obtain the better stability compound. So we focus on the compound called CDD for further study. However, we have to do another proof. Is it possible for CDD to convert to curcumin in our biological fluid? So I test the stability of CDD in human plasma. Into CDD in human plasma, the CDD actually going down along the time and release curcumin going up. That means curcumin can release, CDD can release curcumin in human plasma. Please note that in our body, the human plasma has so many esterase enzymes, and esterase enzymes are actually responsible for the hydrolysis of ester prodrug of CDD and give curcumin as a bioactive compound to exhibit the biological activity. So this is the first paper that we published on the synthesis and the physical chemical property evaluation of the prodrug of curcumin. Later on, we try to identify the metabolism of CDD in human plasma. We want to know what kind of enzyme are actually involved in the hydrolysis of ester prodrug CDD to curcumin in human plasma. So if we go to the next slide, we confirm the in vitro metabolism of CDD in human plasma. So not only human plasma, we also work in rat plasma, dog plasma, and human plasma. We determine the kinetics, looking at the rate constant and the half-life of the hydrolysis of CDD to curcumin. This is more on the physical chemistry point of view. So then eventually using the approach, what we call the indirect approach, using several kinds of inhibitor, enzyme esterase inhibitor, we can determine that in rat plasma, the enzyme esterase that involved in the hydrolysis of CDD to curcumin is the carboxyesterase. But for in dog and human, usually it's go to the pyroxenase enzyme. And these are the key important when we want to develop the prodrug. We need to know the certain enzyme that actually metabolize our prodrug. Then in addition to in vitro metabolism, we perform the in vivo pharmacokinetics. So the next slide is the pharmacokinetic of CDD and curcumin in rat. And we are able to find that CDD actually have higher concentration in plasma compared to curcumin when we give the compound through the IV administration. For the next slide is the oral administration. It actually gives about the same pharmacokinetic profile between CDD and curcumin. The detail can go to the individual publication. So if you want to perform any, if you want to look through the protocol and the result, you can go to individual publication. If you want to do either, I hope it benefit to your thesis. And for the next slide, after the pharmacokinetic study, we look at the biological activity of the CDD in the anti-hepatocellular carcinoma, or actually it's a model represent liver cancer. So what we do, we have to use the CDD or curcumin and do the transport experiment. If you go to the left panel, it's actually the chamber is a 96-well transwell experiment. And then we actually load the compound on the apical side on top of it. And then we collect the permeate, which comes through the bottom of the transwell. And after that, we transfer the permeate, which is the metabolite of CDD, to the HG2 cell. HG2 cell is a hepatocellular carcinoma cell line. And we look at the cytotoxicity of the cell using the flow cytometer. So on the right, top right, is a flow cytometer result. And for this result, we can see that the quadrant 1, we see a lot of normal cell, which is the control, right? But if we use curcumin or CDD, we can
you
see—
have 5 minutes left.
Okay,
you have 5 minutes.
So we can see a lot of cells that have apoptosis, that die because of the apoptosis. And several markers have shown the molecular mechanism on the apoptosis of the cancer cell. Okay, for the few slides left, let's go to the next slide. Is the tumor cancer model. So we can see that CDD actually reduce the size of the tumor in the xenograft model. And this is the benefit of the CDD compared to curcumin. Besides that, we try to do the formulation of CDD using the method called nanoparticle using chitosan alginate. In the next slide. And we observe, we notice that on the right bottom slide, you see the slight green color. That's one show that the nanoparticle can actually enhance the uptake of the CDD into the cancer cell. So we confirm this to the next one using the thermoresponsive nanogel. So we developed some formulation that can apply onto the skin and actually release the CDD and curcumin on the skin. So basically the formulation is stable at 25 degrees Celsius. When we apply on the skin, it's 32 degrees Celsius. Then the formulation release the curcumin and CDD onto the skin. And this is benefit for not only the drug therapeutic, but also cosmetic purposes. So I will go to the end. So to the application of some of our work, we also do the conjugate with the polymer conjugate with curcumin and develop the soluble. The next slide is correct. The soluble curcumin that can be incorporated to the cosmetic product that's compound. And we also test on the stability and solubility. We found that those polymer conjugate curcumin actually better than curcumin in terms of solubility and solubility. So the whole process is a product approach using the small molecule, which is succinic acid, using polymer, which is the polyethylene glycol, and using the polymer, which is chitosan alginate for the nanoparticle to enhance the property of curcumin. And we hopefully in the next few years, we will move our product to the clinical trials for the cancer treatment. So at the end, I would like to acknowledge a number of people that listed on the slide. And since that the time is very limited, so I will not go to the detail of individual researcher. So thank you very much for your kind attention, and hopefully you can get some information on our lab. And I'm glad to answer if anyone may have. Thank you very much.
Okay. Thank you, Associate Professor Padjadjaran for a very informative presentation. We will have a discussion later with all the invited speakers. We will now continue our session to our second invited speaker. Our second speaker is Professor Dr. Yahdiana Harahap. I will read her CV in a glance. Doctoral degree of Professor Yahdiana Harahap were in pharmaceutical science field from the Department of Pharmacy, Faculty of Mathematics and Natural Sciences, Bandung Institute of Technology in 2003. Her field is pharmaceutical chemistry analysis, especially bioanalysis and DNA adduct analysis. Currently, she developed many analytical methods in biological matrices related to bioequivalent studies for quality assurance of drugs. Professor Yahdiana has published more than 70 articles, both in national and international journal or national and international seminar. Please, Professor Yahdiana, time is yours. You have 30 minutes also to share with us your research. I will let you know when the time already 5 minutes left. Please, committee, from the committee or? Okay. Please, Professor Yahdiana, you can start your.
Good morning. Assalamualaikum warahmatullahi wabarakatuh. Good morning, everyone. First of all, I would like to thank the committee for inviting me. It is the third time I give lectures in this seminar, the International Seminar on Pharmaceutical Science and Technology. So I will deliver this is the review about the volumetric absorptive microsampling as a biosampling alternative in clinical trials and therapeutic drug monitoring in the period of the COVID-19 pandemic. This is the outline of my talks. So for the introduction, all of you has known that the data of the COVID-19 in the world from Johns Hopkins University, USA, as of October 25th, 2020, from 188 countries or regions, about 42 million people confirmed COVID-19 cases, and around 2.7% dead, and 67.4% have recovered. And how about in Indonesia? So in Indonesia, until October 25th, the COVID-19 confirmed people about 385,000 and about 3.4% death, and recovered, the recovered cases about 309,000. It is about 80.1%. And I'm sure also all of you know about the symptoms of COVID-19 is moderate to severe respiratory distress, such fever, cough and nasal congestion, fatigue, and also the severe symptoms such as pneumonia and can cause death. And we should do for the prevention, such as washing our hands with soap or alcohol-based hand sanitizer, and maintain social distancing, about 1 to 2 meters, and also wear a mask. And because we know that until now, the medicine and vaccine still in clinical trials. And WHO, since March 2020, has stated that the COVID-19 become the global pandemic. So people should quarantine and work from home, and closing of crowded places, travel restrictions. And also this become a hamper clinical trial and TDM protocols. And because of that, this is the deviation in make the protocol for clinical trial and therapeutic drug monitoring. And if we want to do the clinical trials and therapeutic drug monitoring, we should collect or take the blood or other biological metrics from the patient or the subjects. So this is the biosampling technique to accurate drug quantification in biological metrics for pharmacokinetic studies, pharmacodynamic studies, and toxicokinetics. It is an important aspect of clinical trials of drug development and therapeutic drug monitoring. And we know that there are many kinds of usable metrics, such as blood, cerebrospinal fluid, breast milk, saliva, urine, hair, and also, from all of them, the golden standard of the biological metrics still plasma and serum. To get the plasma and serum, we should take the blood from vein. So this is the venipuncture technique. There are many disadvantages, such as invasive, need medical assistance, we need phlebotomists, and storage condition must be appropriate, and we should take the large sample of volume. So we should do the innovation to the biosampling techniques such as dry blood spot or volumetric absorptive microsampling. What is the DBS or dry blood spot? Dry blood spot, a blood microsampling technique that involves collecting blood on a small filter paper. So after we prick from the finger and then the blood can spot on the paper, many advantages such as less invasive if compared to the venipuncture and small sample volume can be stored and distributed at room temperature. But there's the disadvantages. There is an effect of hematocrit, and this plays a role in determination of viscosity of the blood and can affect the distribution of blood samples on DBS paper, and it can cause the bias in the analysis. And the FAMS, volumetric absorptive microsampling. This is a blood microsampling technique that involves a precise volume absorption of blood using hydrophilic tips. You can see in this slide, this is the hydrophilic tips. After we prick the finger and then the blood, we can absorb the blood with these tools. And many advantages such as less invasive, less taking volume, easy handling, accurate sample volume collection because the volume is fixed, 10, 20, and 30 microliter, and reduce the effects of hematocrit, no need for medical personnel, so the sampling can be done at home. But this is have the disadvantages. This relative, yeah, for the cost, the expensive sampling tool can only be processed once. And because there are many advantages of FAMS, so we can do the sampling at home. If we can do the sampling at home, it can slow down the chain of transmission of COVID-19. And for the analysis, we can combine this biosampling technique with liquid chromatography tandem mass spectrometry or HPLC. So the challenges during the COVID-19 pandemic, because there should be quarantine and do the social distancing, it will inhibit the implementation of clinical trials for drug development and therapeutic drug monitoring. So FDA has stated that the sponsor researchers should consider alternative study workflows during possible disruption. So we need the technological development and innovative approach to increase the productivity of the implementation of clinical trial protocols for drug development and TDM. So this is the sampling alternative. We said that this is FAMS, volumetric absorptive microsampling. And we can analyze it from aspect of clinical and bioanalytical. First, from the clinical aspect, we can observe about the clinical consideration for implementation of FAMS to clinical trials and TDM during the COVID-19 pandemic. The advantages of FAMS in clinical trials: minimally invasive, small sampling volumes, sampling only from the tip of finger or heel, and can be done at home. And it is suitable approach for vulnerable populations such as pediatrics, geriatrics, patients with acute episodic disease, example, epileptic patients, and also will give the patient a comfort to the patient because sampling is faster and easier without the help of medical personnel or phlebotomists and small sample volume. And the sample can store and shipping. We can also save sample storage and shipping costs because the samples can be stored at room temperature, so we don't need the freezer. And delivery in dry samples, patient can collect their own samples at home, so it saves the travel costs. But there's the disadvantages. The sampling tools are expensive and can only be used for one sample. So we should give attention to sample handling and preparation because it requires special expertise to get maximum results. And to anticipate about only one sample, we can use only for one sample. So we should do the sampling more than once. If we compare to the other biosampling technique, venipuncture, dried blood spot, volumetric microsampling, volumetric absorptive microsampling of FAMS, we see for the centrifugation for analyte separation, in venipuncture, we should do the centrifugation to take plasma or serum, but in red blood spot and FAMS, we don't need it. Storage and delivery, for venipuncture, we should store the sample in the freezer, while for DBS and FAMS, it can store at room temperature in dry samples. And for sample preparation, if we want to obtain plasma, we should edit the anticoagulant, while DBS and FAMS, we just cutting the DBS paper or just separate the tips from the handle. And for sampling, venipuncture, more invasive and large sample volume, while DBS and FAMS is similar, minimally invasive, because we can take the blood from finger prick, small sample volume. Second, clinical consideration for implementation of FAMS to clinical trials and TDM for COVID-19. We know that the drug development for COVID-19 should involve high risk to patients, such as pediatrics, geriatrics, and patients with comorbidities, such as cardiovascular, respiratory disease, chronic kidney disease. So we should provide minimal treatment as possible. So the solution is to use FAMS, volumetric absorptive microsampling, because it can increase the patient comfort. Now, we can see here that in the drugs in clinical trials, until right now, there are no one drug for COVID-19 yet. But last week, the FDA has approved the first treatment for COVID-19. This is remdesivir for COVID-19, but still do in hospital or clinic and under the physician responsibility. And also the others, such as favipiravir, chloroquine, and hydroxychloroquine. And it must pass the clinical trials for drug safety, pharmacokinetic profile, drug efficacy, and because some of these drugs have side effects, such as chloroquine can cause the heart problem, cardiotoxicity. And if we know the drug concentration in plasma, that is 20 until 200 nanogram per mil, and also the toxic concentration already stated higher than 3,200 nanogram per mil. So we can do the therapeutic drug monitoring to the patient. So in the implementation of clinical trials for drug development and TDM, the consideration of cost of handling sample is needed, because we know that this test can last for months or maybe years. So funds can be used to reduce research costs because less files, sample cups, and sample preparation tools are used, and no need for anticoagulants, freezers, and centrifuges. So the National Institute of Health, NIH, started a study to measure undetected cases of COVID-19 infection, determine how people have no history of COVID-19 symptoms but have antibodies to the virus. So they analyzed FAMS blood sample from 10,000 volunteers. The study will provide an overview of the magnitude of COVID-19 pandemic in various infected communities without knowing it. And we all know that the test for the COVID-19 swab test is the best test for COVID-19 because it's direct to detect the virus in our body, but unable to detect the previous infection. And another is serological testing. This can detect the antibodies from serum. So the blood take from the vein, this is the venipuncture sample. And then the NIH do with FAMS, and can analyze the antibodies to the SARS-CoV-2 virus. And the use of FAMS is safe, effective, and easy in sampling. Patients can take their own samples from home. And the third factors to be considered in the use of FANS for clinical trials and TDM during the COVID-19 pandemic. The factors affecting volume variation, the FANS sampler tip must not be immersed in the blood because it may absorb excess blood. So we should do the media to socialize the proper and correct use of FANS to the users, to the patient or subjects. And also maybe do the online training forums, video tutorials, and instruction brochures. And we see here that for the volunteer compliance factor, because in Indonesia, the discipline people not underestimate, but maybe rather low. Because of that, we should also must complete and inform consent. The patient is deemed to have complied with the research objectives, rates, and procedures. And because there is no direct control during the pandemic, so we should do many efforts such as periodic control via telephone contact, online forums, or give reminders. So the other aspect for bioanalytical aspect, previous is clinical aspect, and then now the bioanalytical aspect. Yeah. The role of bioanalysis laboratory provides quantitative measurements of drugs in a matrix with suitable analytical methods so as to produce accurate data. In microsampling studies requires validation of the method to ensure the sensitivity and selectivity of the analysis because fewer samples were used. Because of that, method validation must overcome the effects of storage and delivery temperatures, sample homogeneity, hematocrit effects, and stability. This is the bioanalytical consideration for implementation of FANS in clinical trials and therapeutic drug monitoring for fixed volume uptake, stability of analytes and metabolites, homogeneity of sample hematocrit effects, carryover and cross-contamination, and also logistics. For bioanalytical consideration for implementation of FANS in sample analysis, we should to give attention to accuracy and precision, recovery, and matrix effect. So for the fixed volume uptake, if we compare to the conventional sampling such as venipuncture, it is required high accuracy to get an accurate sample. Sampling and handling should be handled by trained analysts. While in DBS, the diameter of each spot is different on different papers because there is the effect of hematocrit. And we usually use the sub-punch technique to get identical dots. While in FANS, because FANS, the fixed volume available in 10, 20, and 30 microliter volumes, but we should also monitor the usage of these tools. For stability of analyte and metabolites influenced by temperature, humidity, sunlight, storage and shipping condition, chemical reaction, enzyme catalyzed degradation. And if we compare again to conventional sampling, in conventional sampling, if we want to minimize the degradation, we can add the enzyme inhibitors, control the sample pH and antioxidants in the collection tube. While in microsampling, if we add these components during extraction, the enzyme and other such as antioxidants, it is allowed degradation when drying the sample. So from the other reference from Kip et al. 2017 about stability of drug miltefosine in FANS and DBS, he compared DBS and FANS. In FANS, the stability is only 1 month at room temperature, while DBS can extend for the stability for 5 months in aluminum bag at room temperature. So for the stability, DBS is better than FANS. So the stability difference analyzed in FANS sample depends on the type of analytes. The stability of analytes cannot be ascertained for a long time at room temperature. And for homogeneity of sample and hematocrit effects, blood with different hematocrit values can affect the blood viscosity, and it will give the variation in the size of spots scattered in the DBS. and can cause the bias in the analysis. Because high hematocrit concentration will give high red blood cell count, and it can be trapped on DBS paper substrate and inhibits analyte desorption process. And it will affect the recovery of analytes and metabolites. If we see the research from Spooner et al. 2015, do the research using human blood and mass with different hematocrit from 20 until 65%, and then absorbed with FAMS. We can see here, this is the human and mice blood with different hematocrites that absorbed by FAMS and investigated in 6 different laboratories. You can see here from this slide, the slope of human blood volume to hematocrit. This is the hematocrit from 20 until 65. And this is the blood volume around 10. So the slope of human blood volume to the hematocrit plot is close to 0. So it means that the volume of blood collected by FAMS has little difference with the different hematocrit blood values. So it can conclude that with FAMS, there are no influence by hematocrit to the analysis. And if we see the percentage of coefficient variance, only 8.7%, that it meets the coefficient variance precision requirements that not more than 15%. And how about the carryover and cross-contamination? If contamination occurs during sample collection, it can cause experimental errors, affect the selectivity and accuracy of the analysis. Sampling technologies should be designed to eliminate or minimize this contamination. If we compare DBS with FAMS, most DBS designs are open like this. After we put or spot the blood on the paper, and then we dried it in the open area, so it will contact to the air and no special protection. And contamination may occur during the drying time. While in FAMS, it is equipped with a cartridge protector. This is the purple, allows drying of the sample in closed condition and can minimize contamination from the environment. For the logistic, FAMS can provide practical benefits because simple sampling, samples can be sent and stored at room temperature, can collect samples by yourself at home, equipped with a collection device and a shipping container. So it is easier for user to use it, and sending samples to the laboratory and become cheaper because it can be done at room temperature. Second, bioanalytical consideration for implementation FAMS in sample analysis. For accuracy and precision, the criteria nominal concentration and percent CV are not more than 15% for QC samples and not more than 20% for lower limit of quantification samples. If we see again that the research from Kim et al. 2017 for the accuracy and precision, the miltefosine drug in FAMS and DBS, the DBS, this is the nominal concentration, 15% for all samples, and for LLOQ 7%, while for the precision, the percent of coefficient variance smaller than 19.1%, while in FAMS is better because the nominal concentration just 10.8%, and the precision, the percent of coefficient variance not more than 11%. So for accuracy and precision, FAMS is better. And for recovery and matrix effect, recovery to ensure test performance and maximize the sensitivity of the method. FAMS reduce the effect of hematocrit. Because of that, it can enhance the recovery. So the use of FAMS tend to have less matrix effect, and the sample volume is less, and processing the sample to extract analytes has fewer steps compared to DBS.
You have 5 minutes left.
Okay. This is the future perspective. So after I have explained before, so this is the future perspective. The use of FAMS for clinical trials of drug development and TDM during the COVID-19 pandemic will be very useful because it's many practical benefits to clinical aspect and bioanalytical aspect. As an effort to reduce in discipline, we can do the training videos or instruction brochures to socialize and train volunteers or patients on how to take samples with friends correctly. In addition, remote monitoring can also be applied via telephone contacts, online forums, or reminders. So in our research group, bioanalysis research group in Faculty of Pharmacy, Universitas Indonesia, is still ongoing now. We want to do the drug monitoring because we know that chloroquine, there's this concentration range, and also it is, we have already the data of toxic concentration. Same with hydrochloroquine and favipiravir and also remdesivir. So chloroquine, hydroxychloroquine, and favipiravir have the cardiotoxicity because it can do QT prolongation. And if combination with azithromycin, it can higher risk of cardiotoxicity. While remdesivir, this is the toxic concentration, it can cause the hepatotoxicity because inhibit hepatic enzyme in our body. So why we do FARMS that I already mentioned before and explain, elaborate why FARMS, because COVID-19 is contagious, a higher risk of transmitting of health workers, invasive by sampling method. So the solution we should find the better by sampling technique that self-sampling can do self-sampling and also easy to use and comfort to the subject or patient. So this is our ongoing research. We want to monitor the chloroquine, hydroxychloroquine, remdesivir, favipiravir levels in fans of COVID-19 patients using HPLC-PDA for chloroquine and hydroxychloroquine, and for remdesivir and favipiravir using LCMS-MS. So we will do the research to 100 subjects for each drug. So 400 subjects we need. We will do the research in Universitas Indonesia Hospital and also the Kemayoran at-risk village. This is the step for biosampling in farms, sampling from finger prick and then drying and extraction. And the conclusion for these talks that the FARMS approach allows for sample collection at a location far from clinic and can be done by the patient at home. This can be applied to the implementation of clinical trials for drug development and therapeutic drug monitoring during the COVID-19 pandemic, which requires quarantine to be held. In the aspect of bioanalysis, FARMS can improve the accuracy, precision, and recovery of the analysis by minimizing the effects of hematocrit. However, FARMS has concern about the stability of the analyte, so that special attention is needed to the stability of analyte in the FARMS sample. So thank you very much for your kind attention. Now I give back the time to the moderators. Thank you.
Thank you, Prof. Yadiana, for very interesting presentation and very related to our current condition now. We finally came to our last speaker on this session, which is Dr. Nyimekar Saptarini. I will read her CV in a glance. Dr. Nyimekar Saptarini is a lecturer in Department of Pharmaceutical Analysis and Medicinal Chemistry, Faculty of Pharmacy, Universitas Padjadjaran, Indonesia. She finished her doctoral program in pharmacy from Bandung Institute of Technology in 2017. Her research interest in enzyme isolation from natural resources. She has published articles relating to pharmaceutical analysis, biochemistry, biological sciences, and environmental science. Please, Dr. Nyimekar Saptarini, time is yours. You have 30 minutes to present your research. I think the committee will, okay. Thank you.
Okay, thank you. Thank you for this opportunity. Today I will deliver our study about crude bromelain of pineapple crown as candidate of herbal immunomodulator. I am Nyimekar Saptarini from Department of Pharmaceutical Analysis and Medicinal Chemistry, Faculty of Pharmacy, Universitas Padjadjaran. Next. Okay, the world experienced as a COVID-19 pandemic, which until now has not been found a suitable vaccine or medicine. One way to prevent the development of COVID-19 in the body is to increase the body immune system. In vitro studies show the bromelain decrease CD4 T cell activation, reduce CD25 expression, activate natural killer cell, increase the production of tumor necrosis factor alpha, interferon, interleukin 1, 2, 6, and granulocyte macrophage colony stimulating factor. Next. Next, please. Okay.
I think it's already next.
Right. Okay. Bromelain is distributed throughout pineapple. Bromelain is a complex mixture of different thiol, endopeptidase, and other components that uncharacterized, such as phosphatase, glycosidase, peroxidase, cellulase, glycoprotein, and carbohydrate. Next. Okay. This study was used pineapple, which collected from Subang District, West Java, Indonesia, one of the pineapple production center in Indonesia. The pineapple crown was chosen because its part has not been utilized properly and become agricultural waste. So the novelty of this study was the bromelain extraction from the pineapple crown, along with its characterization, determination of protease, antioxidant, and immunomodulator activity, then formulation and evaluation of its granule from crude bromelain. Next, please. Okay. Okay. This is the pineapple crown that we collect from Subang district. We extraction with ethanol precipitation because the crude bromelain is a global protein, so it will be insoluble in ethanol. The yield only 0.23%, in that saw the very low protein content in a pineapple crown. The picture below is the crude bromelain was light brown powder, tasteless, and slightly pineapple fragrance. Next, we do the solubility test that saw that crude bromelain forming a colloidal solution in aqueous and insoluble in ethanol, acetone, hydrochloric acid, and sodium hydroxide. That saw the solubility of protein character. Next, please. Okay, next we do the protein qualitative test because we know that bromelain is an enzyme which is a protein. We do the color reaction with nylon, 9-hydrin, and burette test. All the tests show the positive test for protein qualitative. Next, we do the total protein content because we know that we only collect the crude bromelain, not the pure bromelain. So we must know how much protein in this crude bromelain. The result is the absorbance of the casein as a standard is 0.62, which equivalent with 13.03 milligram of milli of casein, which is the protein content in the crude bromelain is 43.44% of protein, and the remaining is the other compounds such as ethanol insoluble compound. Next, please. Next, we do the protease activity for the basic of the crude bromelain activation. The activity we determine from casein degradation to form a tyrosine. Then we measure the tyrosine content. The activity of crude bromelain is 7.72 unit per mil. Our result is higher from fruit and stem of bromelain, which collected from another part of pineapple. So I think our group think the pineapple crown is potential as bromelain resources. Next, we do the optimum temperature and pH optimum. For the temperature, we do the from 40 degrees until 75 degrees, and we have from the result the optimum temperature was 55 degrees Celsius, and from the pH we do from the basic until the alkaline, from the 3 until 12 pH. We have optimum pH is 5. All the character is same like another bromelain from another part. Next, please. Okay, next for the characterization, we do the kinetic parameter. We do the protease activity with different concentration of casein as the substrate. Then we do the calculate the activity. So after that we make the Michaelis-Menten graph. From the Michaelis-Menten graph, we can have the Vmax from the plateau-like phenol region, such as 8.11 unit per milligram minute. Then we calculate the Km from the equation. So we have 1 milligram per mil as a Km. From Vmax and Km, we can calculate the Kcat, and we have 0.03 per second. This parameter, the 3 parameter, is specific for casein as the substrate because the different substrate will have a different kinetic. Okay, next we do the thermal stability. We store the crude bromelain with 2 different conditions at the 25 degrees Celsius and 45 degrees Celsius. You can see that the activity is decreased depend on the condition, and after we calculate, the remaining percent residual activity was only 0.36 for 25 degrees Celsius and only 0.04% for 40 degrees Celsius. Next, we can see that retain approximately 5% activity is reached after 21 days for 25 degrees Celsius and only 4 days for the condition at 40 degrees Celsius. Next, we do the thermal inactivation for another character. After we plot, we see that the thermal inactivator is follow the first order model. So after we know the first order model, we can calculate the rate constant for the 2 conditions, both for 25 and 40 degrees Celsius. Then we can see we can calculate the energy activation. Is it about the 642? This result is higher than other bromelain from another part. Okay, next. Okay, after we know the kinetic, we do the antioxidant activity because we want to know about the immunomodulator. First, we do the antioxidant activity because there is a lot, the protein content is low, only 45%. So the antioxidant activity is have a big number, a big value is about 1,580 microgram per mil. Next, after we do the antioxidant activity, we do the immunomodulator assay. After we finish the ethical clearance and thus calculation, the immunomodulator activity of bromelain is achieved at those 2,000 gelatin degradation unit. Then we calculate and convert this dose to the mice dose as the animal experience. Next. Okay, we do two kind of the immunomodulator assay. First is phagocytosis response. We inject the carbon ink as immune antigen. Then we see there is a carbon absorbance reduction but the reduction is dose independent. So the higher dose is not make a higher reduction. So after that we do the stimulation index calculation, and all the treated group is have greater than 1, so we can conclude that the crude bromelain was immunostimulant, not immunodepressant. Another one, we do the humoral immune response. With this test, we can see the antibody, primary antibody and secondary secondary antibody. The primary antibody is determined at the 7th day after we induce with the red blood cell, and for the secondary antibody we determine at the 15 days after induction. And we can see that the production of primary antibody was prominent as compared to the secondary antibody, and the dose at the 3.12 is microgram per mil is equal to the standard. Next. Okay, after we do the, we know the activity, we do the last step is about the formulation. Because there is only small antioxidant activity, the dose is high, is about 76% crude bromelain as active constituent. Another one is additive. We do the formulation for granule. After we do granulation with wet granulation, we do the evaluation. We follow the compendial method, USP. After we examine, there is a moisture content is 4.1%. The repose angle is 25.70 degrees. It's so the excellent flow for the granule. Then we do the bulk density and tap density. The value is not different big enough. That's how the granule is good enough, because there is a different size. Next, we do the Hausner ratio and compressibility index, because we won't know how about the character of flow of the granule, and from the result we can see that the granule have fair flow character. So after we do the evaluation, we do again the protease activity and antioxidant activity. The protease activity is almost same with the crude bromelain, is 7.73 unit per milligram, and for antioxidant activity, there is a difference with the crude bromelain because there is excipient in the granule. So we can conclude that, next please. Okay, we can conclude that the crude bromelain pineapple has protease activity with the specific character and first order thermal inactivation. Crude bromelain 3.5 12 milligram per 20 gram body weight has immunomodulator activity compared to the standard, which is the immunostimulant, then crude bromelain can be formulated into granule with protease and antioxidant activity. We need a further experience because the granule is not the point, the target. We want to make a capsule or tablet, but because the COVID-19 pandemic, I have to retain our research. Okay, I think this last slide. Okay, that's all from me. Thank you very much. I back to the moderator.
Thank you, Dr. Nimekar, for a very interesting presentation. Now we come to discussion session. As Associate Professor Ponchai already left due to meeting schedule, I will deliver the question from 4 people, which is Mrs. Febrina, and then Mr. Ian, Mr. Yoga, and also from Ms. Drianti to via email to Associate Professor Ponchai. Is there any question for our 2 invited speakers? I'm seeing on the chat room is already like 2 questions for Professor Yahdiana. I think this, okay. First question come from Mrs. Febrina to Professor Yahdiana.
Okay.
The question is, due to its very low volume of the blood collected using the FAMS, is the preparation and extraction method same with the usual plasma or serum samples? That's the first question. I think I finished 2 questions for you, Professor Yahdiana. Is it okay? Okay. The second question comes from Mr. Yoga. He asks, can we measure the biowafer of COVID-19 drugs via FAMS, and how efficient will it be compared to conventional pharmacokinetic measurement? And the last one, I think there's 3 questions for you. The last one comes from Mr. Ian. The analysis step that may be difficult is getting a good extraction recovery from biological fluid. Can you share how to choose the right extraction method based on the structure of the active substance to get the appropriate recovery?
Okay. My face is... Yeah. So, will I answer the questions?
Yes,
no?
Yes, you can answer the question now.
Yeah. So, for Mrs. Sabrina, can we do the low concent? Can we do the sample preparation for low concentration with the same biological and same with the other by sampling? Maybe the question like this. Yeah. So for the low concentration, we can choose the sample preparation. Yeah. There are many sample preparation. We can do the dilution. We can do the protein precipitation. But if you want to do to the low concentration, maybe I give the suggestion we can do for liquid extraction. So after we release the tips to the tube, and then we can add the organic solvent. Yeah, usually we can add the methanol or acetonitrile, but this is for sample for protein precipitation. But if you want to continue to the liquid extraction, maybe it is better because we can evaporate the solvent and then we reconstituted the residue, and then if we do the lower concentration, I suggest that we should use the LC-MS. Because of that, in our ongoing research for chloroquine and hydroxychloroquine, we just do by HPLC. because we already obtained the lower limit of quantification, still higher than 5 nanogram per ml. But if the concentration lower than that, maybe we can use LC-MS. So the sample preparation, we can choose liquid-liquid extraction combination with the methanol or acetonitril to precipitate the protein. Is it clear, my answer? Sabrina? And then,
Sabrina, yes, the answer, yes.
Okay. And the second, Mr. Yoga asked about the BioWave COVID-19. BioWave compared to conventional, I don't understand the question. BioWave, what? BioWave, the COVID-19.
Mr. Yoga, can you I think you can unmute your Zoom, because the question that I read is, can we measure the biowafer of COVID-19 drugs via FAMS? How efficient will it be compared to conventional pharmacokinetic measurement? I think she asked you about doing the bioequivalence and bioavailability for COVID-19 drugs using volumetric absorptive measurement.
Yeah, we can do this for the pharmacokinetic studies. Depends on the drugs. We should know the lower limit of quantification of the medicine or drugs. And also we should try to find the reference about whether the drugs the affinity is most to the plasma or to blood. If it can bind to the erythrocyte, or it can has the large concentration in blood, we can do the FAMS. So I also already have experience to do the pharmacokinetic studies by DBS, dried blood spot of metformin hydrochloride, and it gives the same profiles, pharmacokinetic profile with the conventional biosampling technique, such as in plasma. But in FAMS, I haven't done yet. Maybe later, but I think it can be used for pharmacokinetic for COVID-19 drugs. And we also have already planned about the hydrochloroquine and chloroquine. We want to do also the pharmacokinetic study by FAMS. Is it clear, Mr. Yoga?
Yes, clear. Thank you.
We can do by FAMS. Yeah.
Okay.
Yeah. Because FAMS is better than DBS. We can do with DBS, so it is better if we do it with FAMS.
Oh, okay.
And the third question, the analytical step. For the third question, for the analytical step for obtain the maximum recovery of biological, this is maybe the analytical step to obtain the optimal recovery of analyte. Yeah, how to do the right extraction of analyte and metabolite. It is the same with DBS and also with plasma serum. We should do the optimization of the sampling, sample preparation process. So we should choose, we should select the proper solvent to do the extraction of analyte or metabolite from the biological matrix. So there are many the sample preparations, such as I mentioned before, protein precipitation, liquid-liquid extraction, maybe only protein precipitation and liquid-liquid extraction. Maybe we can do the solid phase extraction. It is difficult for FAMS because the sample is very small. So to do the recovery, yeah, we have already compared in the last year and this year. Also, we already do the FAMS for anti-cancer drugs. And if we compare DBS and FAMS, in recovery, FAMS is better because there is no the influence of hematocrit. DBS influenced by the hematocrit. So if we extract it from the paper, the hematocrit will be retain the analyte or metabolite, while in FAMS, the analyte and metabolite is easier to extract to the solvent. So I think with FAMS, we can obtain a higher recovery than DBS. But with plasma, if we compare to plasma, the recovery in plasma is better. Ya, jadi if we compare from plasma, DBS, and FAMS, plasma still is the best for recovery. But for from DBS to FAMS, FAMS is better, like that. Is it clear, my answer?
Okay, very clear. Thanks.
Thank you.
Okay. Last question for Professor Yahdiana from Mrs. Fabrina again. She asked you to share to us how to handling a sample for COVID patient bloods in the lab.
Yeah. So we already plan to do the research to COVID-19 patients. But until now, we still develop the method. So we just optimization and then validation the method, follow the guideline of FDA and EMA. And then afterward, we have collaboration with Universitas Indonesia Hospital. So we can keep our samples in the hospital in the special freezer there because Universitas Indonesia Hospital also the government in Depok, what is it, West Java, choose the Universitas Indonesia Hospital to be the center of COVID-19 COVID-19 treatment. Yeah, so they already have the facilities for COVID-19, and in our lab we also should arrange the tools and also the facilities so all the analysts or the researchers can save to conduct the research, something like that.
Okay, thank you, Prof. Yahdiana. And maybe it must be done in minimum BSL3 maybe.
Okay. Thank you. This is the question from Mr. Yoga to Dr. Nyemekar. As we know that enzymes like bromelain are very unstable under environmental influences when manufacturing. What is recommended in the formulation so that the enzyme is not degrade during processing?
Okay, thank you for the question. From our research, we know the crude bromelain is still the optimum temperature is 55 degrees Celsius. So I think it can until that temperature until for 55 degrees Celsius, the bromelain is still stable. So I think we can do the wet granulation without heating. It is better than we do the wet granulation with water. So in this research, we use ethanol as the binder for the wet granulation, or we can do the direct compress, but I don't know about because I don't use the direct compress, but because there is a lot of active compound. Bromelain is about 76%. So I don't think the direct compress will be a better result with compared to the wet granulation. I think so. The formulation is okay until the temperature is not higher than 55 degrees Celsius.
Okay, thank you. We still have time because we finished earlier. It's already like 11:30. Is there any question? You can write down your question at chat room if you still have a question. Is there any question from the participant? Okay, there's a question, one for Dr. Nimekar. Is there, from Ms. Drianti, is there any obstacle if we purify the bromelain first?
Okay. If we do the purification, I think the activity will be higher, but the cost will be higher too. So why in this research and for the next research, I only use the crude bromelain because when I compare with the purified bromelain from another research, the activity only higher about 20% compared to the crude bromelain. So I think if we do the purification, there is a long step and more cost compared to the crude bromelain, but the result is not good enough. So I think crude bromelain is fair enough to use as the active ingredient. But there is no obstacle when we do the purification.
Thank you, Dr. Nimekar. Is there any question again from the participant? Okay, if no more questions, so it means we finally come to the end part of this parallel session, plenary session. Before I close this session, I would like to take the conclusion from what the speaker have presented, that an effort have been continually conducted to design and synthesize several prodrugs of natural product, developing biosampling alternative in clinical trials and TDM in the period of COVID-19 pandemic, and finding herbal immunostimulant candidate. This effort has been made to increase the quality of human life. I'd like to say thank you to the speakers for very informative and interesting presentation, and to all participants for very active participation. Hopefully this seminar will give a lot of benefit for all of us. I will back to the MC.
We would like to invite Dr. Alia Nur Hasana to kindly hand over the certificate to Associate Professor Ponchairo Sitisak, PhD, Professor Dr. Yadiana Harahap, and Dr. Nimekar Saptarini. With all respect, we now give the time to Dr. Alia Nur Hasana.
As an appreciation from the committee and also from our dean, this is the certificate for your kind contribution. We will deliver the certificate to your email after the seminar ended. Thank you once again to Professor Yahdiana and also to Dr. Nimekar Saptarini.
Ladies and gentlemen, the next agenda is photo session. We would like to ask all the participants to turn on the camera and take a picture together. We will count to 3 and then take a screen capture of this Zoom meeting. There are 5 slides, so please hold your pose. Now I will count to 3, so please hold your pose until we finish the screen capture. 1, 2, 3. Okay, thank you. The next agenda is parallel session. The parallel session will be held at separate Google Meet link until 1 p.m. The link will be displayed on the screen and also sent via group chat. participant may allow to leave this Zoom meeting and go to this the specific Google Meet.